Hi, my name is David Thomas and I'm the manager of the educational labs here in the Crocker Science Center. Before you get started in your lab course this semester, let's cover a few items that you need to be aware of to maintain a clean, efficient, and safe work area. First, let's talk about PPE or personal protective equipment, beginning with the clothes you wear to lab. Fully closed shoes are required in all labs on campus. Shoes that may enclose your toes but don't cover the top of your foot are not fully closed shoes and are not acceptable footwear. Full-length pants or equivalent clothing that covers all exposed skin down to the tops of your shoes is also required. Leggings do not meet this requirement because they are often too thin to provide adequate protection. Safety glasses and lab coats, and in some cases splash goggles, are also required in all labs. Please be aware that standard prescription eyewear does not count as safety glasses. While you should bring your own safety glasses for this course, lab coats are provided for you. On your first day, you'll find a supply of clean coats in the whiteboard cabinets in the lab. Select one that fits you, and that will be yours to use for the semester. It should be worn any time you're in the lab without exception, but should never be taken into the adjoining classroom. This could transfer substances that are on your coat from the lab environment to the classroom environment. For similar reasons, gloves, equipment, and other lab supplies must always remain in the lab. They should never be taken into the adjoining classroom or other areas of the building. Let's discuss using gloves for a moment. They are on your hands to keep potentially hazardous substances in the lab from getting onto you. You must be thoughtful when using gloves about what you're touching. Rubbing your face because of an itch, pushing back your hair, putting your hands in your pants pockets, or handling personal items with your gloves on are all ways that substances on your gloves can then be transferred to you, potentially putting you at risk. How you remove your gloves also matters. To remove gloves properly, grasp one glove near the palm of the hand using the other gloved hand. Pinch and pull the first glove off. This should invert the glove as it's removed so that the outside of the glove is now inside. Ball up this removed glove in the remaining gloved hand. Slide your ungloved finger beneath the cuff of the glove, avoiding touching the exterior, and then pull that glove off. Again, this should invert the glove. You'll end up with a ball of gloves where the exterior that was used to handle laboratory materials is now encased on the inside, and the clean interior is what is visible. Then you can dispose of your gloves appropriately and wash your hands. Remember, always change your gloves immediately if you get anything on them. Washing your hands is one of the most important safety habits you can develop when working in a laboratory environment. Always wash your hands after removing gloves, before leaving the lab, and before handling personal items. Between lab sessions, you can store your lab coat and goggles in one of the drawers below your lab benches. When you first enter the lab, you'll see that all of these drawers have tape on them. You should choose a drawer that is close to your workspace and does not yet have a name on it. After choosing a drawer, put your name on the tape to ensure others recognize that that drawer is taken. Your TA will let you know if the enrollment in the course is such that it will require you to share a drawer with someone else. Goggles should be placed into a Ziploc bag before being stored inside the drawer, as this will help prevent any contaminants that might be on your lab coat from getting onto your goggles. Coats should be folded so that only the outside of the coat contacts the drawer or other contents of the drawer. This helps the inside of the coat to remain clean. During the semester, if your lab coat gets dirty, either from a spill or just from general use, place it in the provided laundry basket in your lab. There will always be a supply of clean coats in the lab so that you can get a new one when a previous one has become dirty. If you ever can't find a clean lab coat in your size, let your TA know and we'll find one for you. Personal items, such as backpacks and coats, should be stored in the cubbies that are found in the lab. Do not leave these items out on bench tops or the floor, as they present a tripping hazard and can become contaminated or damaged. No food or drink, including water, is allowed in the lab. If you have food or drink with you, please leave it in the attached classroom before entering the laboratory. Before we jump into some items specific to your course, let's go over a few last general safety tips. Never enter and/or work in a lab space unless your TA or instructor is present. When you work in a lab space for the first time, identify the exit locations in the event of an emergency. Your TA should go over the evacuation plan with you on the first day in the lab. Understand the hazards of the substances you'll be working with in the lab before you work with them. One great source of information for better understanding these hazards is the SDS, or safety data sheets. These are available on the web for almost all chemicals and reagents and can be usually found by searching SDS for the name of that substance in a search engine. For instance, if you wanted to look up the SDS for ethanol, in a search engine you could search SDS for ethanol. Now we'll move on to items that are unique to your Biology 1615 course, starting with the organization and location of some of the supplies you'll be using throughout the semester. Some of these supplies and pieces of equipment have a permanent home at your lab bench. Most of these items will have colored stickers to help distinguish what belongs where. All items with the same color of stickers should be kept at the same lab bench. When you're done with lab for the day, be sure to leave your bench neat and organized for those that have lab after you. This includes giving the bench top a quick wipe down with 70% ethanol. Some supplies and equipment at your bench may need to be restocked from time to time. For instance, pipette tip boxes will regularly need to be replaced. There are sets of drawers against one of the walls at the back of your lab where empty tip boxes can be exchanged for sterile full boxes. Place the empty tip box with the green insert rack still inside in the drawer labeled for empties, and grab a new sterile box out of the appropriately labeled drawer. Note that these are organized by the size of the pipette tips. Solutions, centrifuge tubes, toothpicks, swabs, and other items are located on open shelving in the back of the lab below the bench top. The top shelf will typically contain the new sterile supplies, separated into liquid media on one shelf and solid supplies on the other, while empty or contaminated materials should be placed on the bottom shelf directly into the autoclave pans. You'll be using several different types of media plates throughout the semester for doing culture work. A supply of each of these different media plates is kept in the glass door fridge in your lab. Each shelf within that fridge is labeled with the different media plate types to make it easy to find what you need. However, you should always confirm that the plate you're grabbing to use is actually the type you intend to use. Remember, hundreds of students use this fridge, and it is possible that things end up in the wrong place occasionally. To help avoid confusion, each bag of 20 plates is labeled with the name of the media, as well as a set of colored stripes that are unique to that media type. For instance, LB plates are marked with 2 black stripes, and this information is on the outside of a sleeve of LB plates. Every individual LB plate also has these 2 black stripes. When you go to grab a plate from the fridge, use previously opened bags first before opening a new bag. Empty bags can be thrown into the biohazard bins found throughout the lab. Last, let's talk about waste disposal. At the end of most lab benches, you'll find signage to help you know where to properly and safely dispose of different kinds of waste. Reference this chart when needed, and always remember that you can ask your TA if you're not sure. On your bench, you'll find a plastic beaker for used toothpicks and pipette tips. As these become full, carefully dump them into the large red biohazard sharps containers found throughout the lab. The large cardboard bins throughout the lab are for non-biohazard sharps only. Things like non-contaminated broken glass go in these. They should not be used for disposing of biohazards or normal trash. When disposing of items into the non-sharps biohazard waste bins, keep the bins at less than 1/3 full. There are many of these in the lab, so if one is already 1/3 full, please use another bin until the full one is emptied. These are typically emptied once or twice a week. Please pay special attention so as not to put pipette tips or other sharps into the non-sharps biohazard waste bins or regular trash cans. Doing so puts our prep staff and custodial staff at a high risk of being cut or stabbed with contaminated sharps. Treat cotton swabs as sharps for disposal purposes. Remember that you can always ask your TA, instructor, or other lab personnel if you have any questions about the laboratory. Thank you for your attention and doing your part to maintain a clean and safe lab environment for everyone.